So welcome back students. Now let's come to the last session of docking, and you can see that docking can be done in different ways. Either you can have a protein and a ligand docking, or you can have a protein and a protein docking, or you can have a protein or a nucleotide docking, and you can find that in all of these dockings there would be different interactions which could be possible between the interacting molecules. There could be electrostatic forces of interaction, there could be electrodynamic forces, there could be steric forces, there would be solvent related forces, and all of these are in such a way that the steric hindrances between the interacting molecules should be aimed, okay, so that there would be no much of entropy, okay, and there should be minimum binding energy which has been used. And of course there should be also hydrogen bonding as well as hydrophobic interactions between the two molecules, the two interacting molecules. So keeping all this in mind, we can see that the different stages of docking mainly first includes the target or the receptor selection and its preparation, and after that you identify the ligands with which you can interact it, then you make them both interact by a process called docking, and you check the docking results. And the typical docking workflow, it mainly includes, I have just depicted the same thing in a different way: the target selection, the target preparation, then ligand selection, the ligand preparation, then docking, and evaluating the docking result. Now what do we have to do in the target identification or the biological target? Of course you have to identify what is the target, then after that you have to make a three-dimensional structure of that. If the 3D structure is available in the PDB database, you can just take it as such, okay, or else you have to construct your 3D structure using data available, and most probably in most of the cases it is already available in the PDB structure because PDB database. And then after that you have to do a preparation, it should be processed. Now see example, when you get the 3D structure, sometimes they might be getting it by some X-ray crystallographic studies or various other techniques can be used for the to get the 3D structure. So sometimes what happens, certain heteroatoms, that is other than its native atoms, various edX can be linked to the atom through the 3D structure. So you have to remove these heteroatoms, okay, and at the same time energy minimization also should be done, because we want to check always in the biological systems what the process is happening only in the lowest energy process, or in minimized energy only, that the biological target will react with the ligand. So we do not know how we got the 3D structure and how this 3D structure was being prepared and placed in the PDB database. So if energy accumulation was being carried out in the 3D structure when it was being constructed, you need to remove it. So ultimately by preparing the biological target, by removing its heteroatoms as well as by removing its energy, we are trying to make it is becoming a biologically active and a stable molecule. Then you should also know what is the active site of that particular biological target, because there is no point in saying that the ligand comes and binds to the unimportant part of a protein. It should always bind to the active site of the particular biological target, only then it becomes relevant. So you have to find which is the active site, and we will have to specifically check if the ligand is binding to the active site. So knowledge on the active site of the biological target is also important. Now coming to the ligand selection and the ligand preparation, okay. Now the ligands that we have, either you can get it from different, if you know the different databases, you can get it from PubChem, Sync and all that, or else you can use some softwares called the ChemSketch or ChemDraw, and you can draw the chemical or the ligand which you have. Now if you have a chemical compound and you know the chemical nature of it, you can draw it using a software, and after that the ligand as well as the active site of the biological target are being specified, and they are being docked to each other, okay. and the scoring function generates a score depending on which the best fit ligand is being selected. And when you go to look into the ligand selection, something else also I need to say, I already told it, the ligands that we select should be having the least binding energy. And they should also follow another rule, that the ligands should also follow the Lipinski's rule of 5. This is of much importance. Now, what is Lipinski's rule of 5? The Lipinski's rule of 5 states that there should be, if a orally active drug has no more than one violation of the following criteria, only then it should be selected as a best drug. What are the criteria? Let's see. It should not more than that is not more than 5 hydrogen bond donors should be present in that. That is, it should not have more than 5 hydrogen bond donors within it, and not more than 10 hydrogen bond acceptors in the drug molecule. The drug should be a molecular weight under 500 Dalton, and the octanol water partition coefficient should be less than 5. So, if these rules are being obeyed, then we say that it follows the Lipinski's rule of 5. So, the ligand selection depends upon these criteria. That is, only if it follows the Lipinski's rule of 5, or if it is having a least binding energy, then we select that particular ligand. And coming to the softwares, there are lots of softwares which are available online, which we can use for the docking function. Now, Sanjeevani, which was released by IIT Delhi, Schrodinger, which is another paid software, then Gold from the University of Cambridge, UK, Autodoc from the Scripps Research Institute, GemDoc, Hex Protein Docking, Gram, etc. These are various other softwares which have been, some of these are paid and some of them are, now Autodoc and all that, they are being, these are being free softwares which are available, which we can do by ourselves, the docking studies. So, I guess you got an idea about what is the process of docking. You take a biological target, you take a particular ligand, and you make sure that the ligand binds with the biological target with its least binding energy, and you also check whether it follows the Lipinski's rule of 5, that is, the number of hydrogen bonds formed between these two molecules should be less than 5, and it should have a molecular weight less than 500 Daltons, and whatever it is. And also there's other features, that the octanol water coefficient should be less than 5 and not more than 10 hydrogen bond acceptors should be present in the molecule. So, the same principles are being used in all of these softwares, but each software has having different types of algorithms and all that which has been used in it. So, with this we come to an end on molecular docking. Of course, the practical side is not being covered yet, but you got a theoretical understanding about what is docking and what are the softwares used for docking. Thank you for now.